Hello everyone, and thanks for joining us today for our live webinar, Electrophysiological ex vivo assays for compound testing in acute human brain cortical slices. My name is Diane Royer. I am the marketing manager at Neuroservices Alliance, and I will be moderating today's webinar. For those who already attended our webinars, this event is part of a new webinar series that will focus on showcasing our members' innovative assays and technologies. Every webinar will be presented by one of our neuroscientist experts, and they will all follow today's format, 20-minute presentation, followed by 10-minute questions and answer. If you have any question at any time, please submit them in the question box that is located on the bottom of the GoToWebinar panel. Also, this session will be recorded and you will receive the recording after the live session. However, I strongly encourage you to stay until the end, as it is the occasion to have our speaker answer your questions live. Before starting, let me introduce Neuroservices Alliance in a couple words. We are a consortium of CNS CROs specialized in vitro and in vivo electrophysiological and behavioral studies. We deliver endpoints facilitating decision-making to our clients for their CNS drug discovery programs. We value direct and efficient interactions with our clients, and we rely on a team of more than 50 PhD scientists. Today's speaker is Julien Artinian. Julien is a patch clamp engineer at Neuroservices Alliance member Neuroservice. Hello, Julien, and I will let you take the lead from here.
Thank you, Diane. Hello, everyone, and thanks for joining us. Today, I'm glad to present you a new model of studies available at Neuroservice for compound testing, the acute human cortical slices. During preclinical development of drug discovery programs, extensive effort is usually spent in research using rodent preparations. Indeed, many properties of human neurons are recapitulated in animal preparations. However, important differences remain and can severely limit their utility. For example, specific transcriptional regulation occurs in human cells, and this can be assessed using neurons derived from human pluripotent stem cells, the famous iPS cells. But like other culture neuron preparations, the organized network configuration at work in the brain is lacking. Indeed, the human cortical network displays an amazing level of development compared to rodents. Acute cortical slices offer an ideal preparation, allowing compound testing at synaptic and neuronal scales in human ex vivo. This is achievable within an environment that decently preserves the specific network architecture and the broad variety of cell types observed in the human brain. In today's webinar, I will first introduce the methodology supporting acute human cortical slices assays and describe the preparation. I will then share with you a snapshot of our continuously growing database of membrane and firing properties of human cortical neurons, illustrating different endpoints that can be measured for compound testing. Finally, I will present a study case in which we used the retigabine as a reference tool compound to alter those membrane and firing properties. The achievement of this project is the fruit of a collaboration between Neuroservice and the Service of Neurosurgery at University Hospital of Bordeaux-Pellegrin, led by Dr. Guillaume Panchet. The donors that make this model possible are patients with pharmacoresistant temporal lobe epilepsy. This means that all the available pharmacological treatments failed and that surgical resection of the epileptic focus is the last outcome. They are around 38 years old in average, with a male-female ratio of 45-55%. They sign an informed consent form for tissue donation, and all the experimental procedures that we perform are approved by the Hospital Ethical Review Board. The human brain samples that we collect come from temporal lobectomy surgeries, here within the pink area in the illustration, at a rate of around once every 2 weeks. Occasionally, hippocampal tissue may also be available in the case of a medial temporal lobectomy. The sample collected is distal to the core pathological tissue, which is kept by the hospital for further analysis. Therefore, it's not of diagnostic value, and hence, we do not use it as a model of epilepsy. The transportation time from the surgical area to the lab is below 15 minutes door to door. During this time, the sample is placed in a protective oxygenated solution kept between 2 and 4 degrees Celsius. These different aspects allow us to work with healthy brain tissue of good quality for recordings and make this model relevant for compound testing on human targets. Once arrived at the lab, the tissue sample is immediately placed in fresh ice-cold oxygenated protective solution for dissection and trimming. The aim of this step is to get rid of the edges of the sample that were damaged by the surgical excision. As a scale, the yellow square is around 1 centimeter and represents the tissue block obtained after those trimming steps. The typical 1 cubic centimeter large tissue block is then ready for slicing on a vibrotome. We can typically generate between 15 and 20 slices, even more if needed. Here is an illustration of one slice, where the different layers of the gray matter are visible by eye. Please note the importance in volume of the white matter, probably the most striking anatomical difference with rodent brain slices. The cortical slices are then transferred into an interface chamber, which ensures the best conditions of oxygenation for recovery and holding before recordings. This slide shows microscope images of acute human cortical slices. On the left, a low magnification picture in the infrared DIC condition. It illustrates the clear cortex lamination with the pia matter at the surface, superficial and deep layers of the gray matter, and a small portion of the white matter here at the bottom right corner. In the middle are high magnification images of layer 2-3 pyramidal neurons. The one at the bottom shows the contact with the pipette electrode during the recording. You can see the typical features of pyramidal neurons, namely the triangular shape of the cell body, the thick and prominent apical dendrites, and several basolateral dendrites. On the right are fluorescent images of a pyramidal neuron. This is achieved by injection of biocytin during the recording and post-hoc interaction with a fluorophore. Now, I'd like to show you the typical recordings and measurements that we perform in the preparation I just described. The type of recording that we achieved in human cortical neurons, and that I will discuss today, is the input-output protocol. It is performed using the patch clamp technique in the whole cell configuration and in the current clamp mode. It consists in incremental injection of square steps of current, here at the bottom, through the recording pipette. This triggers depolarization of the cell membrane up to a certain amount, called rheobase, which triggers the firing of at least one action potential. For higher amount of current, the neuron fire an increased number of action potentials. Another way to illustrate this protocol is the more classical sweep display, in which one sweep corresponds to a given current step injection and the associated voltage response. Then the sweeps of interest are superimposed. In this example, the current injection go from negative minus 200 picoamps to hyperpolarize the cell membrane, here the response in blue, up to positive 1000 picoamp. The red trace here is the single spike firing response to the rheobase current injection, here 200 picoamps. The black trace illustrates repetitive firing in response to twice the rheobase current, here 400 picoamps. This type of recordings allows visualization and the measurement of some parameters and to compare them across different conditions. For example, the cell type. Here's the typical firing behavior of classical pyramidal neurons. Now you can see the firing behavior of another classical type of neuron, the so-called fast spiking interneurons. These GABAergic cells are able to generate and sustain hundreds of action potential per second in response to lower amount of current compared to pyramidal neurons. Sorry. Counting the number of spikes or firing rate and plotting it against the injected current gives a frequency current plot, or the so-called input-output curve. In black is the firing profile of a pyramidal neuron with a typical rheobase around 200 picoamps. The firing frequency increases progressively with the injected current up to reach a saturation at the highest values. In comparison, the firing profile of fast-spiking interneurons is plotted in red and illustrates obvious firing differences between these two cell types. Therefore, this protocol is an assay for intrinsic excitability, mostly independent from synaptic inputs, allowing comparison between distinct groups and perfectly suitable for compound application and testing. But this assay allows much more. Thanks to our talented software developer, Sabergraph, we use a custom-made script in Igor that allows automatic spike detection. The software then extracts many different parameters at different scales of membrane voltages. Some are analyzed at subthreshold levels, such as the resting membrane potential or the input resistance. Others are related to the action potential waveform and analyzed at a single spike level, like its threshold, amplitude, or half width. And some other parameters are more associated with repetitive firing of trains of action potentials, such as the interspike intervals or the adaptation ratio. These parameters are all electrophysiological signatures of pharmacological targets, such as ion channels. Some of them manifest a kind of one-to-one relationship with its biophysical manifestation. It's, for example, the case of the hyperpolarization SAG amplitude, which is a clear signature of the HCN channels. Other targets will contribute with other entities to a mosaic of electrophysiological manifestations. And this is our job to help you pinpoint the most relevant readouts for your favorite compound. For example, the activity of the Kv7 channels can influence different parameters that can be monitored over time and pharmacological conditions. This slide is an illustration of the library of parameters that we can measure across different conditions. For example, here comparing two cell types, pyramidal neurons in black and interneurons in red. We can study their properties at the membrane level or at the level of single spike waveform and repetitive firing scales. I will not go through the description of all those graphs. This is just to illustrate our analytical capabilities by extracting the most amount of information from these precious neurons. Yet, I pointed out some of the electrophysiological signatures of Kv7 channel activity with a large or small red Kv7 tag for major or secondary contribution, respectively. In this last part, I will present a study case in which we monitored some of these Kv7 channel electrophysiological signatures in response to their agonist retigabine. The KCNQ gene codes for the expression of the voltage-dependent potassium channel type 7, the so-called Kv7 channels. Their activation triggers an outward potassium current called the M current, which produces hyperpolarization of the cell membrane. The M current has unusual properties for potassium conductance. It's a low voltage conductance, so that a small fraction is activated at rest in pyramidal neurons. It's activated by depolarization to peak around -20 millivolts, and it has slow kinetics and does not inactivate. This is well visible in the trace over here, where the current injection depolarized the cell membrane. This triggers the late activation of Kv7 channels, which produce the typical sag, reaching a plateau due to the fact that Kv7 channels do not inactivate. Finally, these channels deactivate slowly when the cell membrane repolarized, producing the medium after hyperpolarization called MAHP. Due to these properties, Kv7 channels are major contributor of the firing rate of a neuron, acting like a brake. That's why it was used as an anticonvulsant drug in epilepsy until 2017 in Europe under the name Trobalt or Potiga in the US. This slide describes the experimental flow that we followed in this study case. 3 types of protocols were applied. First, a test pulse performed regularly ensures the monitoring of the patch quality and the cell health. Then, an input-output protocol, the very same I just described, in which we deliver incremental current steps injections. And finally, a 2-pulse current injection corresponding to the Rio-based currents, eliciting a few number of spikes, followed by a current step of twice the Rio base that triggers more sustained firing, referred to as Rio base 1x and 2x, respectively. The 2-pulse protocol is applied every 20 seconds for 5 minutes in a vehicle condition, and then for 4 blocks of each 15 minutes each under retigabine application, increasing concentration following a log scale. At the end of each liquid period, an input-output protocol followed by a test pulse are applied before moving on the next retigabine concentration. A couple of neurons were time-matched recorded in vehicle only to ensure the stability of firing over time. Here is an example of one pyramidal neuron recorded under vehicle condition for an hour. The first trace of each block is the voltage response to the injection of a Rio-based current. The time course of the corresponding firing rate is plotted at the bottom left as Rio-based 1x. The second trace is the response to twice the Rio-based current, and the time course of corresponding firing rate is plotted below as Rio-based 2x. On the right is the input-output curve, the number of action potential fired in response to incremental current injection. Please note the great stability of firing in vehicle condition over time, visible on traces, on time course graphs, and in the input-output curve. After an hour, retigabine was applied at 100 micromolar, the highest concentration tested. This drastically and rapidly decreased the firing rate for any amount of current injection. With the same layout, this slide presents an example neuron on which we applied increasing concentration of retigabine after a vehicle control period. Retigabine did not change firing rate for any amount of current injection when applied at the lowest concentrations. This is visible both on the traces on the top and in the graph below. However, when applied at 10 micromolar, here in orange, retigabine started to decrease firing rate at Rio-based 1x, 2x, and as well as in the input-output curve, reaching a maximum effect at 100 micromolar here in red. The next slide shows the average data for an arm of 6 neurons. The graphs show the mean firing rate and standard error at Rio-based 1x, 2x, and the input-output curves. You can see that retigabine starts to decrease firing rate from 10 micromolar to have a maximum effect at 100 micromolar. This is particularly visible for the highest values of injected current. Several arms were performed corresponding to a set of 4 increasing retigabine concentrations among the 5 concentrations tested, ranging from 0.01 micromolar or 10 nanomolar up to 100 micromolar, for a total of 16 recorded neurons. The 2 first analyzed parameters with a major contribution of Kv7 channels were the firing rate at Rio-based 1x and 2x. They both show a dose response profile to retigabine with an IC50 of 8.1 and 10.8 micromolar respectively. Looking at other parameters, you can see that only 100 micromolar retigabine significantly increased the Rio-based current value and decreased the resting membrane potential, but without any dose-response relationship. This mitigated result might be surprising regarding the resting membrane potential, especially. Indeed, retigabine decreases this RMP in other cell types and preparations, such as in heterologous systems and cultured neurons. These systems have much more depolarized RMPs, around -60 and even -50 millivolts. At those voltages, an important fraction of Kv7 channel is active, making retigabine much more efficient. However, human cortical pyramidal neurons from acute slices display resting membrane potential values around -75 millivolts. At these voltages, Kv7 channels are practically inactive. These results indicate that RMP and Rio base might not be the best endpoints of Kv7 channel activity for compound testing in acute human cortical slices. This is an illustration of how human tissue may have specific characteristics compared to other preparations from rodents. Other electrophysiological signatures of Kv7 channels were analyzed. For example, the latency to fire the first spike after the onset of the Rio-based current injection, which is modulated by Kv7 channel activity. Retigabine application delayed the occurrence of the first action potential in a dose-response fashion with an IC50 of 10.3 micromolar. Kv7 channels can also modulate the fast after hyperpolarization, occurring immediately after the spike, which prevents the firing of another spike during a certain time window, the so-called refractory period. Retigabine increased this fast AHP with a dose-response relationship with an IC50 of 13.8 micromolar. Other parameters, for example, determinants of the action potential waveform, such as threshold, amplitude, or half width, were not affected by retigabine application at any concentration. This makes perfect sense since those AP waveform features are not electrophysiological signatures of Kv7 channels. This negative data indicate good specificity of the compound on other potassium conductance, as well as a monitoring of the recording quality. For you to have a better idea of the feasibility of running compound testing studies in acute human cortical slices, I'd like to specify that this study comprised 16 validated neurons from 6 patient samples and took 12 weeks to be accomplished. The study case using retigabine as a reference tool compound in acute human cortical slices revealed that the firing rate constitute the primary endpoint for Kv7 channel pharmacological modulation. The latency to spike and the AHP may also be considered as secondary endpoints. Indeed, all those parameters follow a dose-response profile upon retigabine application with an IC50 value around 10 micromolar, which is very close to what is found in rodents. Rheobase and resting membrane potential were not identified as suitable endpoints in acute human cortical slices, contrary to studies in rodents. To conclude, I'd like to point out that our lab in the site of Bordeaux is a specific platform of Neuroservice dedicated to studies in human tissue. The assays we use allow us to evaluate many endpoints depending on your target of interest. As usual at Neuroservice, we conduct tailor-made studies in close interaction with you. The availability of the human cortical tissue is compatible with usual pharmacological studies timeline. I hope I have convinced you that this preparation may represent a useful new service that we can provide you to help in decision-making in your drug discovery programs. Thank you for your attention, and I'll be happy to answer your questions.
Thank you very much, Julien, for this very comprehensive presentation. Yes, we do have already some questions. First one is, do you provide assays for synaptic activity?
Yes, we can certainly use assays of synaptic activity. We already have beautiful recordings of spontaneous and also miniature postsynaptic currents. This is the analysis is underway, but we do not have data set to present for now. We can also perform experiments on evoked synaptic transmission using electrical stimulation electrodes. That's something we can also perform.
Okay, thanks. Now, what solution is the brain slices kept in during transport and cutting on the vibrotome?
Actually, we use a neuroprotective solution, which, as I mentioned earlier in the presentation, kept cold between 2 and 4 degrees Celsius. It's NMDG-ACSF, and we also this solution is the one we use during the transportation and also during the slicing procedure. We follow exactly the procedure described by Jonathan Ting at the Brain Allen Institute, who developed what he called the extended protective recovery method. And in our hands, it works very well, and we have very good results with it.
Thanks. How long after slicing do slices survive and are usable for electrophysiology?
That's a very good point, and that's exactly what led us toward the very same Jonathan Ting, because in this extended protective recovery method, he published data in which he is able to patch longer than 48 hours after slicing. So in my hands, it doesn't last that long, but we can. It allows us to record at least 30 hours after brain slicing. So of course the success rate decrease. It's not as powerful at the first day, but it's possible, and the recordings are very good quality.
Thanks, Julien. Now, do you always have known which human cortical region you are recording from?
That's a tough point. Actually, in terms of medical point of view, we know that we are in the temporal lobe. We know that we can have, when it's a temporal lobectomy, we know it's what's called the point of the temporal lobe. Functionally, I cannot be certain of the correspondence of what kind of cortex it is. That's something. But I think post-hoc, by communicating with the neurosurgeons and the neurosurgeon team, I think we can provide this information, but post-hoc. I mean, afterwards the experiment, not prior to the experiment. And as I mentioned, occasionally we can access the hippocampus on top of the cortical tissue, and in this case we're talking about medial temporal lobectomy, and in this case it's a bit more easy to know what kind of cortex we're talking about, because then we're talking about entorhinal cortex, perirenal cortex, renal cortex, this kind of cortices like perihippocampal cortex, but it's more rare.
Okay. Now, do you have any experience of CMOS imaging technique for whole slice area?
No, that's not a technique we use. We are dedicated to electrophysiological techniques, so we don't do imaging at the Bordeaux site.
Thanks. Can you do equivalent studies to what you showed for retigabine on the fast spiking interneurons?
I'd love to. I love those crazy cells that fire like crazy. The thing is that, of course, they are much less numerous than the pyramidal neurons. They have a less typical shape of the cell body, so of course the success rate will be lower, because we need more time to find them. But I think that's something feasible. Yes, I recorded some of them. For now, I didn't hunt specifically for them, so I don't have any data set to present you or compound application. But if that's become the main target, I think it will be possible. Yes.
Thanks. Do you have experience with plasticity, LTP, LTD, or field recordings?
Not in human cortical slices. No, that's not something we tried. Of course, yeah, we can think about developing some assays in this preparation, but that's something that we never tried yet. In human, of course, we have the experience in rodents.
And also, how similar are the preps from different donors?
That's a good point. Actually, can be very variable. Several parameters can affect this variability. I would say first the size. Of course, the size can be very different. So of course, when we have a large tissue sample, the dissection is much more easy, and we can more easily find the core of the sample be sure to be far away from the edges that are very damaged by the resection. So this is the easiest configuration. For tiny samples, then it's of course it's more complicated, but it's still doable. I noticed that like in rodents, the age matters a lot. When the patient is pretty young, then the dissection is easy, the recovery is easy, there are plenty of neurons surviving. For older patients, it's not the same. And I would say also that the severity of the pathology can also affect the tissue, and in some brain tissue sample, it's clearly evident that the pathology was very severe. And the most striking element that pinpoint this aspect is the presence of blood in the brain tissue, and this makes my life much more difficult. So yes, the answer to make a short answer, yes, it's variable from different parameters.
Thank you, Julien. So now a bit longer question. The main effect on action potential firing is observable at very high concentration of retigabine. Is this concentration specific to Kv7, or it affects other channels at high concentration?
I would say that very high concentration, I would not agree with that, because the IC50 values is 10 micromolar. So it means that at 10 micromolars, we reached half of the max effect, which is pretty decent. And it's very comparable, as I mentioned earlier, to what is measured in rodents preparations in acute slices. So I would not say that we see the effect at very high concentration. But then to answer the last part of the question, we know that retigabine might have off targets. This is the case with other potassium channels. I read something about KV, I don't remember, another potassium channel, let's say, and this one is blocked by retigabine and is involved in apoptosis, so it might contribute to a neuroprotective effect, but only at longer exposure. So we are not talking about brain slices experiment. We're talking about patients taking the drug for a long time. Something else that has been reported in brain slices is that the retigabine might promote the GABAergic synaptic transmission. So we might have a complementary effect at the synaptic level, not the intrinsic level, so that an increased inhibitory tone might contribute to reduce the firing rate. And I do not know at what concentration of retigabine they observe this effect.
Thanks, Julien. Now, what is the flow rate of perfusion you maintain during the recordings?
I use exactly the same flow rate than in rodent preparations. So I would say something between 3 and 5 milliliters per minute. Okay.
How confident are you regarding what specific cortical layer you are recording from? And are you able to label the cells you record for post-hoc identification of specific cell types? For example, parvalbumin positive, somatostatin positive, etc.
There are different layers in this question. So first of all, regarding the layers, I would say that sometimes it's very obvious. The lamination is very clear, and it's so we have dark at the DIC imaging. We can see a dark layer at the middle of the gray matter, which corresponds to the layer 4. So what is above is layer 2, 3. What is below is layer 5. When you find the pyramidal neurons. In human, we can also distinguish layer 2 from layer 3, mainly by the size of the cell bodies of the pyramidal neurons. So in most of cases, I'm confident about the layers. Sometimes it's a bit more difficult, I have to admit, but I systematically measure the distance between the cell body and the pial surface. So we can also segregate the different recorded cells by this distance. So not defining not arbitrary, but making categories layer 2, 3, layer 5. Excuse me. But just segregating according to the distance to the pial surface. Regarding the postdoc analysis of the cell type, we can do easily filling the cells with biocytin during the recording. This gives us this post-hoc revelation of this flow of this molecule, and this gives us access to the complete morphology of the somatodendritic compartment, I would say. So this helps a lot in terms of identify the cell type. For subtypes of neurons, subclasses of neurons, and we're talking about GABAergic interneuron subclasses, then we do have the possibility to do a post-hoc reslicing of the slices to perform immunostainings against the different peptides such as parvalbumin, somatostatin, carotinin. But this needs to be discussed specifically project by project.
Thanks. Is the protocol you use for cutting and patching from acute human brain slices published for reference?
Yes, not by myself, but yes, as I mentioned in the presentation, he's not the only one. But Jonathan Ting performed an extensive amount of work in this prep. He's at the Brain Allen Institute. He has a website, specific website, open access, open source, in which he shares all his methods and recipes for every solution, slicing and recording solutions for different conditions, and he compares the acute human cortical slices versus rodent slices. He published a Jovi paper, so everything is in video. Yeah, so he's not the only one. I also know someone is University Szeged in Hungary, and is doing recordings of fast spiking interneurons in human acute slices. So he published his method to achieve this preparation. So yes, that's something that is published by different groups.
Okay, thank you, Julien. Just before we go on with the next questions, just to let know everyone, we still have more questions to go. For those who have to go, please, you can do so, and you will receive a recording with all the response to the questions. But if you have more time to stay, please feel free to stay. Now the next question, Julien, can you perform post-hoc mRNA analysis on the patched neurons?
We do not have the capability to do it ourselves, but this needs to be double checked, but I'm pretty confident we can extract the RNA, or not extract it, but keep the electrodes, for example, and flash freeze them and then send them somewhere else to analysis. But this is something that needs to be specifically discussed, again, project by project. But so we don't do it ourselves, but I think this is something we can consider. There's something, excuse me, Jan, there's something, another aspect I need to add on this subject is that because of the contract we have with the hospital, we cannot do RNA sequencing that will allow the identification of the patient, because this is anonymous and we do not have the right to know the identity of the patient. I think we're talking about legal aspects. And so the contract we have do not allow us to have access to this information, which is totally normal. And so so this needs also needs to be discussed with the hospital to consider if the gene of interest is capable of identify the patient.
Yes. Okay. Now, if you keep slices overnight for a second day, do you keep in mmDG ACSF in 4 degrees after first day of recordings?
No, not at all. So the method I use is, so I use a specific ACSF for recovery and holding, for the second part of the recovery and the holding. And so this ACSF has a lower sodium concentration and is supplemented with many stuff that helps for the cell to survive, like ascorbic acid, pyruvic acid, HEPAS, this kind of things, and other. So I have 2 interface brain brain slices chamber, and every 12 hours roughly I make a new recovery or holding ACSF solution fresh, and I use the second interface chamber, and then I transfer delicately the slices from one chamber to another for them to rest in a fresh solution. And the first chamber is then thoroughly clean and ready to serve for the next round. But no, it stays in ACSF, not in NMDG, and it stays at room temperature.
Thank you, Julien. Also, due to size differences of pyramidal neurons across age, do you measure capacitance as well?
I measure the membrane properties such as membrane resistance or capacitance. I do see very much an important level of variability in terms of the size of the cells of the pyramidal neurons. I would say mostly in terms of layers. So layers 2, for example, the pyramidal neurons are very tiny, very tiny, and they have the capacitance, for example, around like below 100 picofarads. So which is comparable to what we see, for example, in GABAergic cells, which are smaller. And for example, in deeper layers, in layer 5, then we can see what we call giant pyramidal neurons with capacitance of that can go up to 400 or 500 picofarads. But to answer the first part of the question, I never or I didn't look specifically at the age dependence in terms of size of the pyramidal neurons. So I cannot answer this part of the question.
Thanks, Julien. Now, are you doing also voltage clamp with human brain slices? And how much access resistance do you have usually in the slice?
So the first part of the question is yes, as I mentioned earlier. I do not have a specific data set to present, but I do have recordings, voltage clamp recordings, mainly spontaneous synaptic activity. In terms of access resistance, I would say that this is exactly the same general principles than works in rodents preparation. Namely, the lower you are, the better you are, for sure. The more, if it's constant, it's better. The idea, the rule I like to follow is that the series resistance should not exceed 20% of the membrane resistance. Sorry, 10% of the membrane resistance. So for example, in very small neurons, it's much easier. In large neurons, then the things are different. But in the counterpart of it is that in larger neurons, and especially in giant pyramidal neurons, then you can afford to record with very large pipettes, of course, and this gives you usually a very nice access resistance. But that's something we monitored and follow over time, and then the recordings are discarded when the cell resistance become too high or too variable, like in rodents.
Thanks, Julien. Sorry, just a couple more. Are cells recorded at room temperature?
Not necessarily. Oh, yes, we are very close to room. Actually, I record at 25 degrees Celsius. So it's not. I do not heat much to reach like in other papers, for example, at around 30 degrees. I just use a heater, a solution heater, to make the temperature stable, and so I'm always at 25 degrees. So I would not say room temperature. I would say 25 degrees Celsius.
Thanks. Are some cells spontaneously active, and can you perform a study to look at the effect of compounds on spontaneous firing rate?
That's an interesting question. Let me think about it. It happens. Well, I would not say that's something that is very frequent. Also, I have to say that I do not stay much long time in the Geiger seal configuration, in cell attached configuration. I go whole cell quite rapidly. And whole cell, I would say that not many cells are active spontaneously. I would say that I've seen maybe one or two, and it was difficult to know if it was because the recording was bad, because we know that when the recording is bad, usually the cell depolarize and fire, or if it was because of like a feature of the cell. But I also know that many cells can be spontaneously active and firing, but this feature can be lost once we break in, actually. So that's why I was talking about the cell attached configuration, and maybe if I would stay in the cell attached configuration, I would see some of them, but I would not say that it's something striking. No, it's not a striking characteristic of the human cortical tissue. Okay,
just two last questions. First, what is the access resistance that you get, and is this stable throughout the recording duration?
I think I already answered this question, Jen. My bad. I respond no, that's fine. That's fine.
And then so last, what is the success rate when patching neurons in human brain tissue?
So if we are talking about pyramidal neurons, I would say in average, I can get 2-3 validated neurons per session. And it's an average. So of course we can have crazy sessions with 6, 7, 8 neurons per session, and others where it's much more difficult. So on average, we can talk about 2-3 validated neurons per session.
Okay, thank you very much for answering all these numerous questions, Julien. For now, we don't have any more questions, so I don't know if you have anything to add, Julien, or we can terminate the presentation.
It was my pleasure to present everyone these results and to answer the questions.
Okay. Well, thank you very much, Julien, and thank you everyone for joining us today. This is the end of the webinar. Please, just when you close the windows, there is a very short feedback survey that will open up. We appreciate your feedback, and you will also receive an email within an hour with the recordings, the full recording of the session, and the opportunity to follow up and ask more questions if you would like. Thank you very much, everyone, and thank you, Julien.
Thank you, Jen.
Bye-bye.